Thank you. It's really a pleasure to be back. I spent a long time here, and I'm still a Terp fan. I actually, in my suitcase, I have a Terp 2002 National Championship. I was going to wear it, but I figured it would obscure the tie. So, let me just find, here we go. The title of my talk is Traumatic Brain Injury. It's all about balance. We study traumatic brain injury because it is the leading cause of death in children and young adults. It is an enormous economic burden on the health system, with approximately $7 billion in aggregated costs. That is direct expenditure together with injury-related work loss and disability. Traumatic brain injury causes substantial cognitive impairments in humans. And last but certainly not least, little is known about the causal mechanisms mediating these pathologies. The central hypothesis that our lab works under is that cognitive deficits in animals and humans with traumatic brain injury are due to pathological alterations in GABA and glutamatergic function. These alterations then precipitate regional imbalances between excitatory and inhibitory synaptic transmission, causing frontolimbic system dysfunction. In order to induce our injury, we use a classic model called the lateral fluid percussion injury device, shown here, and it looks like something out of the Spanish Inquisition. We do this exclusively in mice. All the mice come in, and we give them a craniectomy. Basically, we drill a hole between lambda and bregma and off the midline. That's why we call it lateral fluid percussion. Then we seal on a lure lock, basically the male part of the end of a syringe, over the craniectomy with dental cement, and then we allow the animal to recover. The next day, the animal is hooked up to a high-pressure tube, which is attached to this fluid-filled cylinder. A piston is then dropped from a predetermined height. It strikes the piston, causing a fluid wave in this cylinder, and injects a small amount of fluid in between the intact dura, the membrane surrounding the brain, and the cranium. If we have a tear in the dura or we get herniation or anything like that, the animals are not used. And I believe in all the data I'm going to talk about, we've given the animals about a week to recover, and all the slice work I'm going to talk about was taken from the ipsilateral side. And for our controls, we use something called sham animals, which get the craniectomy, all the anesthesia. We hook them up to this device. We just do not release the piston. Okay. Now, here are some of the symptoms of traumatic brain injury. You have an increased risk of seizures, cognitive impairment, and changes in impulsivity and mood. Furthermore, we are very interested in cognitive impairment. And under cognitive impairment, you have alterations in language, attention, memory, executive function, and mental flexibility. Furthermore, our lab is really interested in memory. And memory can be broken down into 3 different types. You have semantic memory. This is how you know you're in Maryland or what the capital is of Maryland. I wrote here episodic-like or spatial because episodic memory in rodents can be a form of spatial memory and working memory. And I'm going to tell you about experiments dealing with episodic or spatial memory and working memory. Now, for normal function in the brain to exist, you have this delicate balance between these 2 systems: excitation mediated by the neurotransmitter glutamate and inhibition mediated by the neurotransmitter GABA. And under normal conditions, you can think of this as a nice balance between a seesaw. However, network excitability can be affected by altering one system, inhibition or excitation, or both. What I mean by that is if you wanted to increase net synaptic efficacy or network activity, you could ramp up glutamate, you could decrease GABA, or a combination of both. And conversely, to decrease excitability, you could ramp up GABA, decrease glutamate, or a combination in both. All our experiments start with various behaviors. In this case, this is called contextual fear conditioning. And we ran this behavior where the animals either went through sham surgery or injury. We gave them 6 days to recover. And then we take both the injured and sham animals and you put them in this box. You put them in the box, they sit in the box for 3 minutes, and then we deliver a shock through the floorboard. And then we let them sit in that box for an extra 30 seconds. We take them back, put them back in the animal colony, bring them back the next day, and we put them back in the box, and we assess their freezing behavior. What I mean by that is we watch them for 5 minutes, and every 5 seconds we make an assessment, are they moving or not? And the only movement they can have is the chest can move up and down for respiration. And we quantitate how much they freeze. Here's the freezing from the sham animals in green, which they freeze about 40%, and the injured animals freeze significantly less. Now, we made that craniectomy right above the cortex that overlies the hippocampus. And this behavior involves the hippocampus. So it's not surprising that the injured animals freeze significantly less. Now, I want to convince you that traumatic brain injury is actually a circuit phenomenon. So after the behavior, we actually prepare brain slices from the animals. We bring them over to the lab, we anesthetize them, we take the brains out, and then we put them on a vibratome, and we cut brain slices. This is the hippocampus shown here, and our experiments start by sticking a stimulating electrode in the afferent pathway, the incoming pathway, into the first region of the hippocampus called the dentate gyrus, which I've signified here with DG. We then put an extracellular electrode in the dendrites. Okay, all the cell bodies are shown here, and the dendrites move up this way. So we're putting this extracellular electrode in the dendritic region. This is very similar. We're basically doing EEG in the slice. Now, as we start to stimulate, okay, we increase the stimulation on the stimulating electrode, and we record the extracellular, what's called field potential or excitatory postsynaptic potential, shown here, and we look at the size or the slope of it. What you can see in the open circles, you have, these are slices from sham animals that have recovered for a week after their operation. As we increase the intensity or the stimulation on the stimulating electrode, you can see that response goes up and it grows, but then it plateaus. And that's very important because this area works like a filter or a gatekeeper for this area. And it does this because the granule cells here, the principal cells of this area, are very hyperpolarized. That means that their resting potential is very, very negative, and they do not like to fire action potentials. Furthermore, you have lots and lots of both what we call feed-forward and feedback inhibition. So lots of GABAergic activity, which keeps everything under control. Notice when we do the same experiment in slices from injured animals that we've allowed to recover for a week, now the curve is amplitude shifted above, meaning for every stimulation, you get more bang for your buck. So you have an increase in network activity or net synaptic efficacy. We went up and did a similar experiment in area CA1, the output of the hippocampus, where we're stimulating the axons coming from this area, CA3. And again, we put an extracellular electrode in the dendritic region, which is called stratum radiatum. Now, as we increase the stimulation, notice here are the slices from sham animals, again, allowed to recover for a week. And now the same exact experiment from slices from injured animals is amplitude shifted below. So the first take-home message is even though the hippocampus is one unit, different things are going on in different regions. Now, when we do this experiment, we're just superfusing with artificial cerebral spinal fluid in both systems. Excitation and inhibition are intact. Now we wanted to see what happens if we look at inhibition. So we use a different type of recording technique where we use something called patch clamp recording. We bring an electrode down and we seal it onto one cell. So we're recording the activity of all the synapses that actually synapse onto that one cell. And we've thrown in blockers to block glutamatergic transmission. So you throw in APV and CEQX to block AMPA and NMDA receptor activation. And we also throw in a poison called tetrodotoxin, which blocks action potentials. And what you have left are what we call miniature inhibitory postsynaptic currents. Okay? And this is due to the spontaneous release or the spontaneous fusion of the presynaptic vesicles that have GABA in them. And then the GABA goes out of the vesicle and it diffuses across the synaptic cleft, and it binds to postsynaptic receptors. What you can see here in the sham, you have lots of activity. This is a continuous recording for about 4 seconds in this cell. And you can see that you have lots of activity of different sizes because this cell has lots of synapses, and the synapses are of different sizes. Notice what happens when we do the same experiment with injured. First, you have a decrease in the frequency of these events, meaning that you have less number of events. And if you analyze those events shown here, they're significantly smaller. So the dentate, as I showed you in the previous slide, has an increase in overall excitability, and inhibition is compromised. Now we go up and do the same experiment in CA1, and you could hypothesize, because in this area, I just showed you in the previous slide, that has a decrease in excitability, so you may hypothesize, is inhibition augmented? And that's exactly what we see. There's no change in the number of events or the frequency, but the amplitude, the size of these events, is larger. So, the central questions that we're going to talk about today are, does lateral fluid percussion affect spatial and working memory? Is the frontolimbic circuit affected by lateral fluid percussion? And finally, how do circuit alterations affect frontolimbic behaviors? So, I mentioned before that the dentate works like a filter. It's also known that hippocampal processing plays a central role in memory formation and retrieval. The dentate is thought to facilitate memory encoding by distinguishing incoming patterns of neuronal activity from previously learned patterns. That process is called pattern separation. And the dentate can perform this pattern separation because, as I said before, only a small fraction of the dentate cells are activated. What I mean by that is that they fire action potentials by a particular input pattern. And that process is known as sparse coding. What I mean by that is you have a lot of activity coming in from the entorhinal cortex into the dentate, but the dentate working like a toll booth or a filter, then the output is this sparse, clear output that then moves it to the next area, which is area CA3, and then it goes on to area CA1. Now, in order to look at the role of the dentate, we used a modified behavior of spatial object recognition called pattern separation. And the way we do this is we bring the animals in and we put them in an arena and we let them habituate. Every time we put them in the arena, they're in there for 6 minutes and then they're returned back to their home cages for 3 minutes. Then after habituation, we have 3 bouts of training. And what I mean by training is we put them in the arena with 3 identical objects that are set up in this diagonal way in the arena. After the last training, then 24 hours later, we're going to move one of these objects to a new location. We move them from this corner up to this corner. And then we assess, can the injured and sham animals discriminate the moved object? And that data is shown here. And we create this discrimination index. We do that by seeing how much time the animals spend with object C in the last training over how much time they spend with object C once it's been moved. You can see down here that the sham animals can discriminate the displaced object C, but the injured animals cannot. And this behavior involves the dentate and area CA3. Now, this is another experiment where we took slices, and here's the dentate, and here's area CA3. We're stimulating over here in the perforant path. We've stained these slices with a voltage-sensitive dye. It gets into the membrane and senses the voltage across the membrane. Now I'm going to show you a movie of what it looks like. So we're going to stimulate, and this is sham over here, and you're going to see the activity. Red means you have lots of depolarization, and here's what it looks like in the injured. So as the next depolarization comes in, you'll see the activity goes along the granule cell body, but nothing gets through, right? The filter is working very nicely in the uninjured animal, or the slice from the uninjured animal. But in the injured, you have much more depolarization, and with subsequent depolarizations, the filter is failing, and you have activity getting through the filter into the next region called area CA3, which is very seizure prone. We believe that this phenomena is why you have a decrease in seizure threshold. Now I'm going to show you another movie, and now depolarization is going to be warm colors, yellow and orange, and when you hyperpolarize, we'll show you blue. Again, we're stimulating in the perforant path. Here's the stimulating electrode up here. This is a sham in an injured slice. And what I want you to see is that in the sham, once again, you get nice depolarization, but very little gets through. This is all where it's supposed to, where all the granule cells are, but nothing gets through. But in the injured, you have significantly more depolarization, and it goes all the way down and through the filter, because the filter is failing, into here, into area CA3. Now, these are just frames from the movie just showing you that there's much more depolarization in the dentate, shown here in red. The peak is significantly different. You can see the dark line is the mean, and the shaded area is the 95% confidence interval. And yet, then we looked further downstream in area CA3, and you can see that there's lots more depolarization, significantly, compared to the sham, to the slices in sham. Now we went through and did some more physiology. Once again, we're going to stick a stimulating electrode in the perforant path, the afferent pathway coming into here, and we're going to stick an extracellular electrode in the cell bodies of where these granule cell layers are, shown here, and again in area CA3 in the cell body layer. And we're going to tweak the system by giving it 2 stimulations, and we're going to see how in the second pulse do they fire these action potentials. And because we're recording them extracellularly, we call them population action potentials, because it's the whole group is firing an action potential. And first we looked at how many slices will respond with a population spike in area CA3. First, you can see in the sham slices, only about 35 to 38% of them will fire a pop spike with this second pulse, but in the injured population, that goes up to about 60%. Now, we're drawing here the input-output curve for this second pulse, and you can see the pop spike here. Because we've tweaked the system, in the dentate, there's no difference between sham and injured. You can see here in sham is the pop spike, and down here in injured. But in CA3, they very rarely fire an action potential with the first pulse, where am I? Here. But after injury, they're all firing it, shown here in the open circles. Now, furthermore, we were very interested, this graph shows you, now we're looking at the first pulse to show you that after injury, we increase the excitability in the dentate. So the sham is shown here, but in the injured, shown here in the open circles, they're firing more action potentials even with the first pulse. This is the same graph I just showed you with the second pulse. And then if we cut the axons going from dentate to CA3, there's no second stimulation. There's no firing because we've killed the ability for the cells to fire. Now, we further went ahead, and this is the structure again. We're stimulating in the perforant path. Here are the granule cell layers. Here's my granule cell. Here are the dendrites where we were recording before. And then here's the axon that's called the mossy fibers that synapses on area CA3. And we were interested in where in CA3 was the hyperexcitability or the increase in network activity coming from. So in CA3, the axons synapse on an area called stratum lucidum, right here underneath the cell body layers, and yet in CA3, every axon that comes out of a cell body then sends a collateral to its neighbors. And those collaterals then synapse down here in stratum radiatum. So we can put our recording electrodes either close to the cell body in stratum lucidum, shown here, or then move them to stratum radiatum. And now what you can see, if you look down here in the group data, when we look at the injured population in stratum lucidum, now we're recording where the axons come in, the injured population is significantly more excitable. And yet when we do that in stratum radiatum, shown here, they are not different. So the excitability in CA3 is coming from the axons from dentate into CA3. Okay, so just to summarize this first part, the dentate becomes more excitable. Here's my little seesaw showing you that there's more excitability, more glutamate, less GABA. And in CA1, you have a decrease in excitability. There's definitely more GABA or augmented GABA, and we've also done experiments to show that there's decrease in glutamatergic activity. Because the activity or the network activity in CA1 is diminished, this is the general output of the hippocampus, and that output is diminished. Now we're going to switch gears, and when my first neuroscience graduate student came to the lab, he says, I don't want to work in the hippocampus. I want to work in the medial prefrontal cortex. And he gave me some very good reasons why. So he told me that, or it is known, that the prefrontal cortex receives dense projections from the hippocampus. It is also a site distant from the injury. So here's the craniectomy. The fluid pulse is going here, and the hippocampus sits right underneath the cortex that's going to feel that fluid pulse. And the prefrontal cortex is far away from that injury. And finally, the prefrontal cortex is involved in working memory dysfunction. So, as happens in our lab, everything starts with a behavior. So, this is a T-maze, and we put the animal in the starting gate. Notice that when we open the starting gate, the animal can only go up and turn left, as he does. Okay? We also keep all these animals at 80% of their body weight, so they're hungry, so they'll do things for food. Then we put him back in the starting gate, and now when we let him go, he has a choice to either turn left or right. But only if he turns right this time does he get a peanut butter chip. Okay, so they have to learn to alternate. If they turn right, then they got to turn left, and vice versa, all the time. So then we take a cohort of animals, and we take them through this test, 10 trials a day for a week. Okay, and they get pretty good at this. They can do this about 75% of the time. If we have what we call a stupid animal, they can't get to 75% of the time, then we just don't use them. So they've gone through, and here, after a week of training, half the animals undergo sham procedure, and the other half undergo injury. and then we run them in this test for the week afterwards. What you can see here is that for the whole week, the sham animals continued to improve, but the injured animals get worse, okay? Then what we did is took this data and looked, what do they look like for the first 3 days versus the last 3 days? We wanted to know, are they improving over the week? And the answer is, the injureds are not. So they have an effect for the whole week, and they're not getting any better. Now that we have a behavioral problem, we're going to look at the circuitry. So I said before, you have a monosynaptic pathway that comes from area CA1 into layer 2 of the medial prefrontal cortex. Layer 2 then sends afferents, or axons, down to layer 5, and from layer 5 you have a polysynaptic pathway back to the ventral hippocampus. So now we're going to stick a stimulating electrode in layer 2, and we're going to stick an extracellular electrode in layer 5, and we're going to go back and create our input-output curve. What you can see here is this is the field excitatory postsynaptic potential in a slice from the sham animal in green, and a field excitatory postsynaptic potential in a slice from the injured animal. And this dip down is the fiber volley, which is the presynaptic action potential. And here we're plotting the amplitude, okay, how big these are, versus the amplitude of the fiber volley. And you can see the injured population is in red, and the area ellipses are the 95% confidence intervals. So once again, like area CA1, you have a decrease in excitability. Now we're going to switch our recording and bring a patch electrode down to, because we wanted to look at the afferent activity onto these cells. And we do this by starting to look at excitatory synaptic transmission. And we do this by throwing in a blocker to block GABA-mediated transmission, which is bicuculline. And this is what you can see. What happens when you look at, these are action potential dependent events. They're called spontaneous excitatory postsynaptic currents. Every once in a while, they fire an action potential. And when we analyze the data, you can see that you have an increase in the frequency of these events in the injured population, but no change in the size of these events overall in the amplitude. We then throw in our poison, the tetrodotoxin, to block action potentials, which then you're left with the miniature excitatory postsynaptic currents. Again, you still have an increase in frequency with no change in amplitude. Then we shifted over and ran the same experiment, but now we're looking at inhibitory synaptic transmission, and we throw in our blockers to block NMDA and AMPA, and this is what you have left. So now you can see that there's no change in frequency, but both with the spontaneous inhibitory postsynaptic potential, the action potential events, and the action potential independent events are smaller in the injured population. So just to give you an idea of exactly what I'm talking about, you have an increase in the frequency of excitatory drive onto these cells together with a decrease in the amplitude of the inhibitory drive, which would basically leave you with more excitation onto these cells. So we wanted to know, how does that affect the intrinsic excitability? So what I previously showed you, that's called doing voltage clamp. Now we're going to do current clamp. You switch a little button on the amplifier, and now we're going to give you hyperpolarizing, the downward deflection waves, and then we're going to depolarize, so you can see we're firing action potentials. And we're going to look at, in response to a depolarizing stimulus, a positive going wave, how many action potentials do we fire? And there's no difference between the injured and sham slices in action potential frequency, but interestingly enough, the voltage where these guys are going to fire their action potential, called the action potential threshold, has higher in the injured population, and that's important because there's no change in the resting membrane potential. So, in conclusion, you have more excitation and less inhibition drive onto these cells, but these guys, from the slices from the injured animals, have changed their action potential threshold, probably or maybe as a compensation so that they're not firing, right? Because you're dumping too much glutamate on them. GABA is less, so they should be firing many action potentials, but maybe they've manipulated their own membrane or their own action potential threshold so that they don't fire in response to this. So now we're going to move the patch electrode down, and we're going to look what happens in layer 5. So we're going to start again and look at excitatory synaptic transmission, and then we're going to look at inhibitory synaptic transmission, and the end all is that there's no change in excitation or inhibition from layer 2 onto layer 5. And then we're going to look at intrinsic excitability, and now what you have is you have a change in the input resistance. So the membranes you can think of look like a little piece of Swiss cheese. They have holes in them that are channels and stuff, and after injury they have more holes. So the resistance goes down in injured, And the effect of that is when you're firing an action potential, the half-width or the duration of this waveform is going to be significantly less. Okay, so just in conclusion on the medial prefrontal cortex, you have more excitation and less inhibition onto layer 2. The slices from the injured animals shift their action potential threshold. There's no change in the afferent information from layer 2 to layer 5, but these cells become leaky. Now I'm going to change gears and we're going to go back to the hippocampus. I told you earlier that we have these changes in excitability in the hippocampus. So we were interested, could we actually measure that? So we grinded up the tissue, took it down, and we ran HPLC, high-pressured liquid chromatography, on it to see if we could measure changes in the concentration of GABA and glutamate. Well, I don't know about you guys, but in my lab, a lot of times things don't work out so well. And there was no change in GABA and glutamate at all. And I shelved the data. And then I went to a colleague's university to give a talk, just like I'm giving today. And one of those colleagues is sitting right here, and he's become a very good friend of mine. And we were sitting at dinner, and with our other colleague, we had a discussion. And they said, have you looked at precursors for GABA and glutamate? So I came back, went right in the lab, and opened it up. And lo and behold, out of all the amino acids, 3 specific amino acids were significantly less on the injured side. And these 3 amino acids are valine, isoleucine, and leucine. And I'm not going to quiz you, but these are essential amino acids, which means you can't get them by diet. You can't synthesize them, so you'll have to get them by diet. You just had lunch, so now you have them in yourself as well. These amino acids, when they get in, I'll just go through this really quick. So when I think of glutamate and GABA, I'm a simple-minded physiologist, so these guys are packaged into vesicles, and action potential invades the presynaptic terminal. The calcium comes in, the vesicles fuse with the presynaptic membrane, the contents, either glutamate or GABA, are released into the synaptic cleft, they diffuse across and bind to their channels, and then glutamate diffuses away from this and is pumped into the astrocyte, shown here. It is converted to glutamine by glutamine synthetase, and glutamine is returned back over to the neuron through a glutamate-glutamine shuttle, or a glutamate-glutamine cycle, and then glutamine is converted back to glutamate by glutaminase. Well, after you have a meal, the situation gets a little bit more complicated. The branched-chain amino acids get filtered a little bit through the liver, but then they get put in the blood, they come up to the blood-brain barrier, and they go through the blood-brain barrier very, very quickly, okay? And you have these low-capacity, high-affinity transporters that actually grab them out of the blood and bring them into the brain. When they come into the brain, they come into the astrocyte, and the acronym that I'm using is BCAA for branched chain amino acid. All amino acids can donate their nitrogen group. These amino acids donate their nitrogen group to alpha-ketoglutarate, and when alpha-ketoglutarate gains a nitrogen, lo and behold, it becomes glutamate. So these are precursors for all of glutamate, and all GABA comes from glutamate. So these are precursors for both the excitatory neurotransmitter glutamate and the inhibitory neurotransmitter GABA. And once you make glutamate, it goes back into the glutamate-glutamine shuttle, and then you can get glutamate over here. And as I said before, if this was an inhibitory neuron, you'd have another step where glutamate is converted to GABA through glutamic acid decarboxylase, and once again, GABA can be released and it binds to a receptor. All right. One thing I just wanted to make notice of is when the amino acid donates its nitrogen, it becomes a keto acid, okay? And the keto acids then are shuttled back from the astrocyte to the neuron, where once again, they can gain an amino group and become a branched chain amino acid. Okay, so then we went back and we ran our original experiments. These are slices from sham animals, and we're doing our input-output curve in the dentate gyrus again. Then we have slices that were from injured animals. Notice you have an amplitude shift above, just like I had shown you before, an increase in network excitability. Now, while we're doing this, we add all 3 amino acids to the superfusing artificial cerebrospinal fluid, and after 20 minutes, this curve in red shifts down to the one in yellow, okay, so we can restore excitability. Now, in the next couple of slides, we've taken this point here in sham, the highest stimulation, and called it 1. And we've normalized all the data to that. You'll see what I mean in a minute. So here is the slices from the sham group. We've normalized it to say it's 1. Injured, the population shows increased excitability or network activity, so it's above the line. And I just told you if we apply a cocktail of all 3 of these amino acids, you can restore excitability. In this experiment, we applied either valine, leucine, or isoleucine, and all 3 of them have just as much restorative power, so there's no synergism in it. Then we also took a non-hydrolyzable branched chain amino acid called BCH. This means that it can't donate its nitrogen. And when we did that, it did not restore excitability. If we bath applied glutamine, that's the substrate to make glutamate. So that restored excitability. And now if we took the 3 keto acids, they didn't restore excitability. They actually made things worse. Okay? Now, we went up to CA1 and did the exact same experiment. So here are slices from sham animals that we've allowed them to recover for a week. And then the injured is significantly decreased, right? Their amplitude shifted below, so a decrease in overall excitability. And then we bath apply the cocktail of amino acids, and lo and behold, we can restore excitability. So think about that for 1 minute. You have opposing shifts in excitability, you give them this cocktail, and magically you can bring everything back to normal. Pretty surprising. Now, we went ahead and did the same experiment. Here's, again, slices from sham animal. Injured is significantly, shows a decrease in overall network excitability. And then if you apply each amino acid, each one is just as restorative as the others. There's no synergism. Going again, BCH, the non-hydrolyzable amino acid, does not restore excitability. Now, in this case, glutamine also does not restore excitability. It becomes an unusable substrate. We don't know what to make out of that yet. And the keto acids also don't restore excitability. Okay, so I talked about some of this data in that table I just showed you a couple slides ago. This is the concentration of the amino acids in sham slices, okay, for all 3. Injured, they're significantly down. And then if we took a cohort of injured animals and we put them on a therapy, we put them on a diet where we put these amino acids in their drinking water for another 5 days, we could restore the concentration of these amino acids in the injured hippocampus, in the injured side where we're delivering the injury. Then we took those animals, or we took another cohort of animals that were injured and we put them on our therapy, and we ran our contextual fear conditioning. We're going to start here because this is exactly the same way I showed you before, where the animals are injured or they have sham surgery. We give them 6 days to recover, and then we train them on day 6 and test them on day 7. Sham freezes about 25%. The injured freeze significantly less. Lo and behold, when we put them on our therapy, we can fix their freezing, right? So we can fix their anterograde, meaning can you learn something new? We also did this behavior when we actually train them. Then we do sham surgery or injury. We give them 7 days to recover, and we test them again. So can you remember what you previously learned in a retrograde manner? Here is sham, animals that were sham. Here are injured animals. They freeze significantly less. And lo and behold, when we put them on our therapy, we can fix their contextual fear problem. So we can fix up both anterograde and retrograde contextual fear impairments. Then we cut slices from those animals. Here's again slices from sham animals. Injured shows an increase in network excitability. When we put them on therapy, they're back to normal. And again in CA1, slices from sham animals, slices from injured animals show a decrease in network activity or net excitability. And when we put them on therapy, we can fix them back up. Okay, now I want to move. We did some experiments on sleep. So when humans get a concussion, and I forgot to say this, how many of you in the audience have had a concussion? Wow, significant amount. And what they tell you to do when you got a concussion? They probably brought you home and the doctor says, don't go to school and just if you don't use your devices and stuff, and just try to just stay quiet, right? That's what people do. So, and also, when you had your concussion, did you sleep more? Yes, I see some yeses and stuff. And so in our animal model, we also have similar effects. So what we did is we actually did some EEG. So we put electrodes through the craniotomy onto the brain, not into the brain, but just on top. And when you do this, you also have to attach a muscle in the back of the neck. Why? Because if you look at the EEG activity when they're awake and when they're going through REM sleep, it looks exactly the same. And you only know the animal's asleep because there's no activity in the neck muscle because he's sleeping. Okay, now I'm going to show you what it looks like. And this is called a hypnogram. So this is when the animals are in a special cage and we have infrared beams that go down so we can see when the animal's moving around or when they're sleeping. This is during the day when the lights are on, right, from 7 a.m. to 7 at night. And what you see is when the animal is sleeping, it fluctuates between 3 states. Wake up here in the green, non-REM, and REM. And they float, but they keep switching between these 3. But notice that in the sham animals, when the lights go off, and rodents are nocturnal, you have these large bouts when they're actually awake and foraging for food, okay? Now look what happens in the injured. So here, when the lights are on, okay, the animals look just like sham, right? They're fluctuating between wake, non-REM, and REM. But when the lights go off, there's no change, right? They still look just like the lights didn't even go off. And now look what happens when we put an animal on therapy. We bring back these long bouts when the injured animals, when they've been on therapy, now have long bouts in the wake. So TBI causes an inability to maintain wakefulness, which is reversed by our dietary or branched-chain amino acid therapy. Now, this is EEG while the animals are going through this. In the wake, and you can see what it looks like. The wake is in green, the red is injured, and then the blue is injured animals on therapy. Now, in your brain, you have endogenous rhythms. One of the rhythms that I want to talk about is theta rhythm. And theta rhythm you've probably heard of. You have a lot of it during REM sleep, and rodents have it when they're running or when they're exploring. And so you can see this really well down here in REM. You have a huge peak, and the theta rhythm is about 6 to 10 hertz. So you see a big peak right here in the green. It's completely absent in the injured, and then when we put them on therapy, you can restore it. You can see it here with the hump over here in green. It's non-existent in the red, but it's back when we put them on therapy. And here it is in green up here. It's missing in the red, but we can restore it. Okay, so then we took these animals and we put them in a novel environment to force them to stay awake, right? And then after they were in the novel environment, we actually took them out and handled them for another hour. So we're forcing them to stay awake. Then we sacrificed them, and we ran immunocytochemistry, and we were interested in a population of neurons that are present down here in the hypothalamus called orexin neurons. They have orexin in them, and they release orexin. Orexin neurons are supposed to be firing a lot when you're awake. It is my hope that as I'm up here talking, your orexin neurons are firing lots of action potentials. So then we ran immunocytochemistry, and we wanted to know, does our injury kill these orexin neurons? And the answer was it doesn't. It doesn't kill the orexin neurons, but we could measure their activity because when they're firing a lot of action potentials, they put out the immediate early gene, CFOS. And then we co-stained them for CFOS. And what you can see, here's the sham levels of CFOS. Okay, so they have a lot of activity. The injured has less activity because there's less co-staining with CFOS. And when we put them on our therapy, we can fix that up. Okay, so we seem to be fixing the sleep problem because we're making these orexin neurons fire more action potentials. Okay, so what I've shown you up to this point is we take injured animals, we give them 2 days to recover, and then we put them on our therapy for 5 days, and then we test them in contextual fear. Now we were interested, what happens if we only put them on for 2 days, 3 days, or 4 days? And that's shown here. So what I've shown you before is if we put them on 5 days, then the animals freeze not significantly different than their uninjured counterparts. But if you put them on for 2 days, they only still freeze 20%, but look at the difference that even at 2 days compared to their injured cohorts that are on water alone. So even though they're not back to normal, they are better than the injured cohort that are only getting water. Let me go back one. Right. Then we wanted to see what is the longevity or duration of the therapy. So we did that by putting either injured animals just on water for 10 days or putting them on therapy for 10 days. And then we had a third cohort where we put them on the diet for 5 days and then took them off for 5 days. And that's shown here. So this is what they look like, and we're doing this all with contextual fear conditioning. So when they're on water, they don't really improve. They only freeze about 25%. So they don't improve over the time for 10 days. But here's the injured on therapy for 10 days. They're back up to 40%. And then if you put them on and then take them off after 5 days, they revert to looking like injured. So at 5 days, there is no durability. I can't tell you yet if we put them on for 10 days or 20 days, will that hold up? Then we did a dose response. We're using 100 millimolar. Basically, that's the highest concentration we could get into the drinking water while they were doing it. And when we did it for 50 millimolar, half the dose, at 5 days, they still weren't back. Okay, now I want to go back. This is our pattern separation behavior. When I showed you that the injured animals could not discriminate the moved object, right? But when we put our animals in our therapy, lo and behold, we can improve their pattern separation, and now they can discriminate the object. Okay, I also talked to you that we have EI stands for excitatory to inhibitory imbalance, and this is what I showed you before. This is the slices from the injured animals. Here's in red is the injured, and green is from slices from sham animals. And you can see there's a decrease in network excitability. And I have to say, I also have to tell you that this slide holds a very, very nice part for me because this was done by my own son. In high school, he came to work in the lab and he learned how to do extracellular recording, and this is all his data. So what he did was, here's the average in injured, and here's the average when we put them on therapy. And really, I'll go through here, and this is easy right here to see. These are the same curves in red and green from the injured and sham animals. And when we put the, when we bath apply the therapy to our injured slices, lo and behold, we can fix it. So our therapy also fixes the excitability in the medial prefrontal cortex. Okay, in the last part of the talk, I'm going to talk about memory. Memory is composed of 3 phases: encoding, which is the initial transformation of an event into a neural representation, and that representation is defined as an engram. Now, maintenance is basically the storage of that neural representation over a period of time or the duration of the engram. And in the final part, you have retrieval, which is the ability to voluntarily bring that neural representation out of storage into the forefront of consciousness. So how do we go about doing this? So this time, we're going to put electrodes into the animal's brain while it's intact. This is called in vivo recording, and we're going to record local field potentials. And we're going to put electrodes all over the hippocampus. We use these very fancy silicone probes so we can record from the entire hippocampus. Now, we're going to go through and do, just like I showed you in the pattern separation, this is classic spatial object recognition. So we put them in an arena, we let them habituate, this time for 30 minutes. The next day, we bring them in through 2 training bouts where we're calling familiarization 1 and 2, with, again, two different objects than we used in pattern separation, but the objects are identical. And each familiarization is 10 minutes each. Then after this familiarization, we move the objects, and we either tested a cohort of animals by looking at them at 3 minutes after this familiarization, an hour, or 24 hours later. Okay, and what you see is at 3 minutes, the injured in the dark boxes, in the sham or in the open, can both discriminate the moved objects. In an hour, the sham can discriminate the object, but the injureds cannot, and when we did it at 24 hours, neither one could discriminate the object. So how do you interpret these results? This is a memory model. And what you have is you have a threshold where you have to get the engram over in order to bring it back. So what I just showed you is that both animals, sham and injured, can discriminate the object at 3 minutes, but not in an hour. So if we think about this is the maintenance impairment, at 3 minutes, right, the encoding is up here, so you have the same peak of the engram, but yet we think that the decay of the engram is faster in the injured population. So at 3 minutes, you're still above this threshold where you can have the engram, but in an hour, injured have decayed faster, or the engram has decayed faster, so that now it's underneath the threshold, so you can't retrieve it, but the sham is still there. And if you look at it at 24 hours, both are going to be below this. However, what happens if it was an encoding problem, meaning that the engram was not as good in the injured animals compared to the sham animals? Then what you would see, at 3 minutes, they could still do it, but now since you have different peaks and they're decaying at the same rate, now at an hour, the injured are going to be below the threshold to be able to retrieve the engram. So we went back and looked at the familiarization 1 and 2, and these are sham animals up here, injured animals up here and sham animals down here for the whole 10 minutes, and the different shading is how often they're within 1 millimeter of the different objects. And what you can really see, just shown here, is for the first couple minutes, the sham animals in blue spend more time with the objects, so their encoding is better. So it appears that brain injury causes a problem in the encoding phase, and that's why they're having a problem with the discrimination. Okay, then what we did is we went through and put a cohort of injured animals on our therapy, and lo and behold, we can fix up their discrimination. They are now able to discriminate in an hour, and lo and behold, we fix their encoding problem. And in the last data slide, or the last 2 slides, this is an example of where we're putting our depth electrodes in CA3 and CA1. We also have them going down now all the way to dentate. And you can see the traces are showing you the electrode. After the animal is done, we check to ensure where our electrodes placements are. here's the one in CA3 over here, and here's the one in CA1. And then what we do is we're looking for the interaction between waveforms or rhythms that underlie memory. We talked about theta before, which is shown down here. This is from 6 to 12 hertz, so you're looking at about 8 to 10 hertz right here, so you go up. And then the other waveform we're looking for, the other rhythm, is called fast gamma, which is about 80 hertz. So you can see in the sham animals, there's a beautiful correlation for the first couple minutes of encoding where these waveforms are interacting. That's called cross-frequency coupling. So if you have theta, which is a low frequency, it's going like this, and the high frequency either sits in the well or the peak, and they interact together. And there's a perfect interaction during the first couple minutes, and then it falls off. Now look in the injured population, okay? You have more of it here, and it does not decay. Now, as a scientist, we're always taught more is better, but not here, okay? Here, there's an optimal range for cross-frequency coupling where the encoding is good. If you have more of it, it doesn't work as well, and if you have less of it, there's not. Autistic kids have more cross-frequency coupling, and so do patients with Parkinson's disease. And that's why we think that they have cognitive impairments. So, in summary, I've shown you that concussive brain injury using the lateral fluid percussion leads to altered behaviors. I've talked to you about spatial cognitive impairment, contextual fear, spatial object recognition, and a modification of the spatial object recognition pattern separation, and a working memory impairment using the T-maze and a delay non-match the sample, or if they turn right, the alternation rule. And we have imbalances in excitation to inhibition in the hippocampus. You have opposing regional shifts in hippocampal EI imbalance. You have inhibition shifts in the appropriate direction in area CA1 in the dentate to contribute to these overall shifts in excitability. And you have increased excitability in area CA3. In the medial prefrontal cortex, you have a decreased network synaptic efficacy. And our dietary therapy ameliorates injury-induced cognitive impairment by restoring EI balance. And it mitigates injury-induced inability to maintain wakefulness, possibly by restoring hypothalamic orexin neuron activation. And finally, it also ameliorates pattern separation impairment and spatial object recognition. And I'll just conclude by acknowledging the people in my lab and my grant funding, shown over here. And thank you very much for your attention.
Sharing this very interesting work on traumatic brain injury. There's one or two questions.
Sure.
Have you determined if there's any foods that contain these BCAA branch chain amino acids?
Yes, so in the meal that
you
you had, they're in lots of stuff. You can get them through, they're protein. You get them in chicken, you get them in fish, you get them in anything. The only thing I don't know is if you eat soy. I don't know if they're in soy, but they're certainly in regular sources of protein. Go ahead.
Applicability to the concussion protocol, such as NFL, would you measure criteria?
So you're asking me in the NFL, can I measure? Oh, you mean? I can't measure them in vivo. Well, so, yeah. I think you're asking me, so what is it that you want me to measure for the concussion protocol? You want us to do EEG or you want us to do?
Well, right now it's pretty subjective.
Correct.
So, but is it concrete?
I see what you're saying. So in answer to your question, there have been studies where people have measured branched chain amino acids in concussed people. You can see a decrease in the branched chain amino acids in the serum, in the blood, after severe, sort of moderate to severe brain injury, but there's also been a study that you can see it in after milder concussion. So you can do that, and our research is trying to get you, instead of having it subjective, to have it objective, and we're working on that. And I forgot to say that this stuff, we've converted this or translated this into a clinical trial that's ongoing on campus. We've made our therapy into a sports drink, sort of, and they have to drink 2 bottles of it. We created a website to test their reaction speed and their memory, and we need about 50 kids, and we have about 17 of them so far going through. So we'll see how it works out. Oh, yeah. So in the clinical trial, we're trying to get them in within 72 hours, and they drink the bottles of this up to 21 days. We have started experiments like what happens if we injure them and give them a week to recover and then put them on. And I think that's where you're going. And so I don't know the answer to that. I think that it'll still be helpful, but if you wait like a month or something, will they recover? We've also shown that our shifts in excitability last out to at least a month, and we have some behavioral problems also out to a month.
Thank you again.
Okay.